Our next question in the physical biology exam is how many sugar transporters are needed to make a bacterium? Okay, and to do this, this is a fun estimate because it will start forcing us to think about the chemical composition of the cell. And by pure thought, we can figure out a lot of things. Okay, so the strategy we're going to use is, let's assume that most carbon is in proteins. Okay, and basically the idea is that if I want to make a new cell, I need to bring as much carbon as I had already inside that cell, right? So we need to figure out how much carbon, how many proteins there are in the cell. Okay, and how are we going to do this? Well, let's think about this. Estimate. So what we're going to do is estimate number of proteins by weight. Okay, so let's start by estimating the weight of E. coli. Okay, so the easiest way I know how to do this is to think about what is the volume of E. coli. Oops. And E. coli is basically, in case you haven't thought much about it, it's basically, on the order of 2 micrometers by 1 micrometer. And basically what I'm going to say is that I'm going to think of this as a little cube of side 1 micrometer, which means that the volume of E. coli is going to be about 1 micrometer cube. Okay, so, or as we saw in the previous vignette, the same as 1 femtoliter. Okay, and what does this tell us? Well, this, if we assume that the density of E. coli is basically that of water, assume, E. coli is more or less the density of water, then this tells us that the mass of E. coli is approximately 1 picogram. Okay, so this is the total mass, but most of it is water, right? In fact, what is the dry mass of E. coli? What would be your guess for this? This in principle should be on the order of 30% of the total mass of E. coli. Okay, so, or about 0.3 picograms. Okay, and what is the total protein mass? Oops. We're going to say that this is about 1/2 of the dry mass. That half of the dry mass is dedicated to proteins, which then tells us that we're at 1.5 picograms. All right, so now that we know the total mass of proteins in E. coli, we can figure out how many proteins we have in E. coli by dividing by the mass of a single protein. And we know what this mass is because we already talked about it. The mass of one protein is on the order of 30 kilodaltons, so 30 times 10 to the 3 daltons, which means that the number of proteins in E. coli is just the mass of proteins in E. coli divided by the mass of one protein, which I'm going to say is on the order of 1.5 times 10 to the minus picogram is 13, right? Picogram is minus 12, minus 13 grams divided by 30 times 10 to the 3 daltons. And what we need to remember is that 1 dalton corresponds to the weight of 1 mole of hydrogen, which is 1 gram. So basically we get 1 gram per mole per dalton. Okay, so this cancels out. What we get here is something on the order of, let's do, here I'm going to do it like this instead. 3 times 10 to the 4. So we get 1.5 divided by 3, that's 1/2, and then we get times 10 to the minus 17 moles, and we know that there's 6 times 10 to the 23 molecules per mole, which means that there's on the order of 3 times 10 to the 6 protein molecules in E. coli. Okay, and this is a super useful number to carry around. Like 1 molecule, 3 million molecules in E. coli, like kind of 3 million molecules per cubic micron. And it's very useful because it allows you to look at data and right away see, is there something suspicious here or not? For example, what I'm showing you here is the result of 2 different studies measuring the copy number of every single protein species in E. coli. The one on the left was done using mass spectroscopy. The one in the middle was performed using microscopy, where they labeled each protein molecule. 1 second, I just lost access. I just lost the mirroring. Let's see if this fixes it. There you go. I apologize. There you go. Okay. So now look at these plots, these histograms, and see, what makes more sense to you, given that you know that there should be 3 million copies of all the proteins in E. coli. You can see that if you add up the numbers in these bars, you could imagine getting to 3 million proteins, but there's no way you're going to get 3 million proteins out of this. And so basically what you see when you compare both measurements is that there's an undercounting by almost a factor of 1,000. And the cells will grow more or less in the same media or similar media. So you can't, it's just the microscopy measurement is basically undercounting. And this is part of the intuition that we talk about when talking about developing a feeling for the organism. All right, so now we know the number of molecules, but we wanted to figure out the number of carbons in the cell, right? So to figure out the number of carbons in E. coli, assuming that most of it comes from proteins, let's do the number of proteins in E. coli, okay, times the number of amino acids per protein, times the number of carbons per amino acid, okay? And we know most of these numbers. We know that there's 3 times 10 to the 6 proteins. We know that on average it's like on the order of 300 amino acids per protein. And to know the number of carbons per amino acid, I had to look it up. I don't know biochemistry enough, but basically on average you get like 5 carbons per amino acid. So times 5 carbons per amino acid, which basically tells me that this 3 with this 3, that's a 10. So 10, and then 10 to the 6, 10 to the 2, that's 10 to the 8 times 5. So this is on the order of 5 times 10 to the 9 carbon molecules, okay? But it turns out that the carbon molecules are imported as glucose molecules. So now we need to figure out how many glucose molecules this corresponds to. And, glucose. And, we know that there's 6 glucose molecules, carbon molecules per glucose molecule. Carbon divided by 6. So we're talking about a 10 to the 9 glucose molecules to make E. coli. Okay, all right, so this is how much we need to bring in to make a new E. coli, right? And these are the molecules are going to be brought in by carbon transporters, such as, for example, PTS1 that transports on the order of 200 glucose molecules per second. Okay, but now we need to remember one thing. It's not just about bringing 10 to the 9 glucose molecules. You need to bring them in the time it takes to make a new cell, right? And what's that time? That's on the order of 10, 20, 30 minutes in E. coli. So what I'm trying to say is that the rate with which I need to bring glucose molecules in E. coli is going to be 10 to the 9 glucose. And if you got a division time of like 10, 20 minutes or something like that, it's on the order of 1,000 seconds. which means that we need to bring in 10 to the 6 glucose molecules per second, okay? But we know that one PTS molecule brings in 200 glucose molecules per second, which means that the number in E. coli of PTS1 transporters is the rate, some more room here, in E. coli that I need of glucose molecules divided by the rate of a single PTS1 glucose. So we're talking about 10 to the 6 glucose molecules per second divided by 200 glucose molecules per second. And so this is on the order of, let's see, it's 1/2 divided by 10 to the 4 PTS1 molecules, which is on the order of 5,000 PTS1 molecules. So just by thinking, we predict that we need 5,000 PTS1 molecules in the cell in order to bring enough carbon at the rate necessary to make a new E. coli. And if you look, the interesting thing is that this 5,000 number is pretty much on the money. So here is some data from mass spectrometry where you can see that they've measured different transporters and as a function of the growth rate. And 5,000, our estimate in red is around here. Is our estimate. So you can see that just by thinking, we can figure out how many PTS1 molecules, how many carbon transporters are necessary to make a new cell, because we were able to figure out how many proteins make up the cell and how many glucose molecules are necessary to provide the building blocks for those proteins.